Hi everyone, today we're going to be talking about cellular respiration by Hunter Polanyi. Cellular respiration accounts for the majority of all energy used by eukaryotic cells, and our goal is to produce maximal ATP molecules in the presence of oxygen, which is aerobic, and our game plan is to utilize the inner mitochondrial membrane to harness the hydrogen gradient and power ATP synthase. So our first step in cellular respiration is glycolysis. Glycolysis occurs in the cytoplasm, and no oxygen is required to start glycolysis. So our first step in glycolysis is we will obtain a glucose molecule from the food we eat, and then in order to break down this glucose molecule, we will have to invest 2 ATP molecules in order to split this molecule. When we split this molecule, our electron acceptor molecules NAD+ are able to acquire electrons to form NADH molecules, which will then be used in our electron transport chains. Also, our ADP molecules are able to acquire phosphates to make 2 ATP molecules each. So in total, 4 ATP molecules are produced. But as you can see at the beginning, we invest 2 ATP at the beginning in order to break down glucose. So our net total of ATPs produced are going to be 2. So all in all, our production during glycolysis is 2 NADH molecules, which we use in the electron transport chain, 2 ATP molecules, which we use for cellular energy, and then 2 pyruvates, or 2 pyruvic acid molecules, which will then go into our bridge reaction cycle next. So our next step in our cellular respiration is the bridge reaction, or a pyruvate oxidation. Pyruvate is oxidized as it goes through the membrane into acetyl-CoA. Pyruvic acid enters the mitochondrial membrane protein and is oxidized into acetyl-CoA with the help of coenzyme A. This is the start of complete oxidation of pyruvate. So our molecule of pyruvic acid enters our cell membrane, and when it does so, carbon dioxide is released, and once carbon dioxide is released from pyruvic acid, it becomes acetic acid. And after acetic acid is formed, our NAD+ electron acceptor molecule will grab 2 electrons from acetic acid, and then our NADH molecule that was formed will go on to electron transport chain, and then our acetic acid and coenzyme A molecule will combine to create acetyl-CoA molecule. Our next step in the cellular respiration is the citric acid cycle, or the Krebs cycle. The citric acid cycle occurs in the matrix of mitochondria, and our reactants for the citric acid cycle are acetyl-CoA, 1 molecule of ADP, 3 NAD+ molecules, and 1 FAD+ molecule. And on our product side, we have 2 CO2 molecules, 1 ATP molecule, 3 NADH molecules, and 1 FADH2 molecule. So during our citric acid cycle, our electron acceptor molecules, NAD+ and FAD, will pick up electrons during the cycle, and then they will be reduced to our electron carrier molecules NADH and FADH2, And then these electron carrier molecules will go on to the electron transport chain, where electrons flow to oxygen and become water. And then our ADP molecule, during the citric acid cycle, will pick up a phosphate and become one ATP molecule, which will be used for energy. Our last step of aerobic cellular respiration is oxidative phosphorylation, or the electron transport chain. So our electron carrier molecules, NADH and FADH2, drop their electrons into the electron transport chain, which then these electrons flow through the electron transport chain, and then our final electron acceptor, oxygen, will take these electrons, and then our oxygen molecule will become water. While these electrons are flowing through the electron transport chain, it powers the hydrogen pumps to pump hydrogen atoms into our inner membrane space. This creates a very high concentration of hydrogen atoms in the inner membrane space and a very low concentration of hydrogen atoms inside our cell. So these hydrogen atoms have to flow from a high to a low concentration, and the way they do that is through our ATP synthase. When they flow through our ATP synthase, it creates a spinning, and then so our ATP synthase molecule will spin, and when our hydrogens flow through and it creates the spinning motion, it allows the synthesis of ADP in a phosphate molecule to create ATP. And then this is an example of chemiosmosis, the use of a hydrogen gradient to drive cellular work and to create the synthesis of ATP. The vast majority of ATP production in cellular respiration occurs in this electron transport chain. Our last step in cellular respiration is fermentation. Fermentation occurs in an anaerobic situation or if there is no oxygen present. So fermentation happens directly after glycolysis, and it is used to keep glycolysis running by oxidizing pyruvate and the two NADH molecules produced by glycolysis. When they're oxidized, they oxidize into alcohol or lactate, which is alcohol fermentation or lactic acid fermentation, depending on the situation. So when they are oxidized into alcohol or lactate, it allows the NAD plus molecule to continue oxidizing glucose for the substrate level 2 ATP production. So fermentation is used only in certain situations where there's a lack of oxygen, like at the end of a marathon, and you only need a small amount of energy. And because in glycolysis there's no oxygen needed to perform, fermentation can happen, and the small 2 ATPs can be used to provide energy for the cell.